Hello students, this is your Dr. Prats and welcome to my channel. Today we are going to start another important series on odontogenic tumors. These are the tumors which arise from either odontogenic epithelium or odontogenic ectomesenchyme. Let's see what is the classification, how the etiopathogenesis works in this case, and clinical, radiographic, and histopathological features of all the important tumors. Let's roll. So, all our presentations are in a questioning format, and it starts with what is the pathology? Why do we need to understand the pathology? The three hows, that is, how did it occur, which is nothing but etiopathogenesis. How to diagnose using your investigating tools, that is radiography and histopathology, and if any other required. How to treat, very important, or how to plan your treatment. Followed by summary of whatever we learned. And lastly, and very importantly, answering multiple choice question. Whenever a cell is faced under any sort of stress, it undergoes adaptation either physiologically or there could be pathological adaptation. So some of the ways it adapts is by demonstrating atrophy, which is nothing but reduction in the size of the cell. Hypertrophy is an increase in the size of the cell. Hyperplasia is an increase in the number of the cell. The size remains the same, but there is an increase in the number of the cell. And dysplasia, which is, there are a lot of dysplastic features which you will be understanding when you're studying the pre-malignant lesions and conditions just before we go to the malignant tumors of the oral cavity. But here you can understand that it is a bad growth. And metaplasia is transformation into another type of cell. So these are some of the ways the cell tries to cope up with the stress which it is facing. So you know that there are some cells which are continuously dividing, and our oral epithelium is one such cell. Others are epidermis, that is your skin, and the GI tract epithelium. So these are called as continuously cycling labile cells. There are also stable cells which have the capacity to divide, but after one cycle they decide to go into a G0 phase or a resting phase, and they retain this proliferative capacity until and unless there is a trigger, they do not do so. So an example for that is hepatocytes. And of course, we have permanent cells which do not replicate after birth, that is your nerve cells and cardiac cells or cardiac myocytes. So this is the cell cycle. I want you all to understand this. And the cells, how they multiply or how they proliferate, because it is important to understand the topic which we are going to study, that is neoplasia. So there are several phases in the cell cycle: G1 or GAP1, synthesizing phase, G2 or GAP2, and of course mitosis, that is the cell division, followed by cytokinesis, where the cell cytoplasm is divided and two daughter cells are formed. So mitosis is what is seen in case of eukaryotes. That is human beings, we are eukaryotes, and that is why I'm explaining the mitosis. There are some checkpoints, that is after G1 and here between G2 and M. So if there is any kind of discrepancy, the cell will not go ahead with the cell cycle. It will not replicate, but instead it undergoes what is called as apoptosis or cell death. So apoptosis is a programmed cell death, where when there is such kind of discrepancies, the cell is killed by the body itself. Okay, so this is the cell cycle. So quickly I'll tell you what happens in each cycle. So G1, S and G2 are together called as interphase, which is nothing but a cell preparing itself to undergo mitosis. S is of course synthesizing your DNA material, okay? And in mitosis, if you see here, so the chromosomes are arranged in the center, so that's the metaphase, and then the spindles get attached to the replicated or the doubled chromosome on each end, and then they pull them to the poles, and the cytokinesis happens after this to produce two daughter cells with equal number of chromosomes. So that's how the cell cycle happens. Okay, so I told you what happens when there is any discrepancy between these two checkpoints, then the cell will not complete the cell cycle. There's another phenomena which you must know, that is the cancer. So how a normal cell becomes malignant is whenever the genes which are controlling the normal cell growth do not take part in their activity, or something has gone wrong with them. They're not functioning normally, basically. So these are proto-oncogenes, anti-oncogenes, apoptosis regulatory genes, and DNA repair genes. So like I told you, whenever there is a DNA damage, the cell functions in such a way that either the DNA repair becomes successful and then the cell proceeds further, or the cell is killed by apoptosis. But when the cancer mechanism is such that it escapes or evades this whole mechanism, and that's how it eventually becomes malignant and continues to grow into a tumor and metastasize, and you know how cancer is such a killer. So with that, we'll start with the neoplasia, which is nothing but an irregular growth of tissue, or irregular proliferation of cells to form a mass of cells. And this can be either benign or malignant. And the benign does not have the capacity to metastasize, whereas when it is malignant, it not only destructs the localized region, but it also has the capacity to invade and metastasize to distant regions. And that is why most of the cases of death by cancer is due to the metastasis more than the primary tumor. So the tumors which we are going to study today are all benign. They are odontogenic in origin, and that is why they're called as odontogenic tumors. So this is a classification of odontogenic tumors given by WHO in 2003. And as you can see, it's broadly classified as benign tumors and malignant tumor. Under benign tumor, there is further 3 classifications based on how the origin is, and also it depicts the histopathology basically. So the first one is odontogenic epithelium without any odontogenic ectomesenchyme. Okay, that's the first one, and these are the tumors under it. I'm going to discuss each one in detail and tell you which are the ones we're going to discuss. And the next is odontogenic epithelium with or without the dental hard tissue formation, and this has odontogenic ectomesenchyme. So it is the interaction between the odontogenic epithelium with odontogenic ectomesenchyme which basically results in the formation of hard tissue. Okay, and the third is mesenchyme and or odontogenic ectomesenchyme with or without odontogenic epithelium. So here the absentee is the odontogenic epithelium, may or may not be present. And under malignant tumor, there is carcinoma and sarcoma. Carcinoma, as you know, are epithelial in origin, and They mainly metastasize by lymph node, and sarcomas are connective tissue in origin, and the main mode of metastasis is through blood. So we will read this as and when we're discussing each tumor, because these are mostly discussed as complications, and for an undergraduate level this is not necessary to read in detail. So I'll just be mentioning it when we are discussing it as a complication, mostly under ameloblastoma. So under the odontogenic epithelium, without odontogenic ectomesenchyme, we are going to discuss ameloblastoma, which is very, very important for you all to know. It can come even as a long essay, a short essay, and several multiple choice questions from this topic. And it is a very aggressive tumor, so as a dentist you have to have knowledge about it. Calcifying epithelial odontogenic tumor. If you remember, when we were talking about the series of cysts of oral cavity, we talked about calcifying epithelial odontogenic cyst, and this is the tumor part of the same, and squamous odontogenic tumor. The next classification, where you see both odontogenic epithelium and odontogenic ectomesenchyme, with or without the dental hard tissue, we have ameloblastic fibroma, ameloblastic fibroodontoma, adenomatoid odontogenic tumor, and odontomes, compound and complex. And the last type under this is the odontogenic ectomesenchyme, with or without any odontogenic epithelium, under which we'll be discussing odontogenic fibroma and benign cementoblastoma. So malignant odontogenic tumors, I told you, are classified as carcinoma and sarcoma, and under that there are mentions of these, and like I said, these will be briefly discussed, not in detail. All these odontogenic tumors have similar clinical and radiographic features. Of course, the histopathology differs, and that's how you can identify or diagnose each one of them. But I'm just going to tell you the most common description of the clinical picture and the radiographic picture, so that it's easier for you when you're studying for exams. All of them are slow growing, which also means that they're asymptomatic, because if the patient has symptom, of course, he's not going to let it grow so big, right? So they're asymptomatic lesions, and they're discovered on routine radiographic examination. Suppose the patient has come for some other treatment or with some other problem, and when you take a radiograph, you accidentally discover it. Or the patient might come to you saying that one of his teeth is absent. So that could be a reason for you to be taking radiograph and then discovering that there is a tumor. These tumors can also prevent the normal eruption of the teeth which is associated in that area, and also can cause displacement of adjacent teeth. And when they're large, they can cause painless swelling of the jaws. So these are like some of the clinical features which are common for all the odontogenic tumors. So let's see what are the common radiographic features. I think this is very simple. It depends on the nature of the tissue. So if the tumor is only of epithelium in origin, then it usually looks radiolucent. And if there is hard tissue formation, of course, you will see that there are radio opacities which can be appreciated on the radiograph. And these radio opacities are basically enamel and dentin which is laid down. And you will see how that there could be even tooth-like structures in these tumors in our coming lectures. And when it comes to whether it's unilocular or multilocular, the smaller asymptomatic lesions are usually unilocular, and the larger lesions are multilocular. Histologically, it depends on the classification, so it'll be easier for you if you remember the tumor falls under which classification, then you will be able to describe the epithelial and connective tissue components. So that's the two components basically, epithelium and the ectomesenchyme in this case. So epithelial components will resemble odontogenic epithelium. So either it resembles like ameloblasts, that's one of the descriptions which we give. And the mesenchymal component will resemble the dental papilla or the dental sac or dental follicle. Because of the interaction between this epithelial and the mesenchymal component, you see the presence of the dental hard tissue. So whenever there is a tumor of mixed origin, that is of epithelial and ectomesenchyme, also make sure that you check for the presence of dental hard tissue. Like I always say, do not get overwhelmed. It's pretty simple. We try to understand how a cell reacts whenever there is stress by atrophy, hypertrophy, hyperplasia, dysplasia, and metaplasia. We also try to understand what is neoplasia based upon the cell cycle and the genes which are responsible to control the cell cycle, or which are the regulators of cell cycle rather. And then we saw the WHO classification given in the year 2003. And I just told you some clinical picture, radiography, and histopathology which are common to these tumors based on which classification it falls under. Treatment part differs based on the tumors, which we will study in the next part of this series. Which of the following are considered as permanent cells is the MCQ for the day. The full form of MCQ is multiple choice question. So this is a stem. Which of the following are considered as permanent cell is the stem. One of these options or the choices is your answer, and the rest are called as distractors. Okay. So tell me which is the correct answer in this. Is it epidermis? No. Epidermis has the ability to proliferate, and it proliferates its entire life. Of course, I hope you all remember what is permanent cell. This does not have the replication potential after the birth. Okay, the next cell I will look at is the hepatocyte. These, yes, they do have replication potential, but they just choose to stay in the G0 phase or the resting phase. What about nerve cell? Yes, they are the permanent cells. They do not have any replicative potential. What about cardiac myocytes? They are also permanent cells. How will you answer this type of question when there is a choice of two correct answers? So in that case, I just want to tell you that there is another type of question which is called as multiple response question. These are also very common in your NBD exams or any other qualification exam. So these are multiple response, meaning you should know both the answers which come under this. There could be like several, there could be like 6 or 7 options, and then you need to tick what are the correct responses under that. So this will help the examiner to understand that you do know all the correct answers which are responses for the stem which is asked. I hope that is clear, students. And if you like today's presentation, and if you liked the way I explained about the multiple choice question and multiple response questions, more such questions are available on my website. The description box will give you a link to my website where you can practice plenty such questions. So these are my references. So Caution is one book which is amazing. I know in many countries, Schaeffer's is still considered as a standard textbook, and it's been edited by 2 Indian authors. There was another book which I thought was pretty interesting, that is by Dr. Anil Ghosh, and Caution still remains my favorite somehow. Like the pictures are amazing. There is always a table which gives a summary of each pathology, which is so easy to remember while studying for exams. So, in case you want to understand through my lecture and then still go back and refer to any of the books, then I suggest you go for this. But because I was teaching you guys, I have included a very well-known book for odontogenic tumors, written by Ricard and Philipson. Thank you, students, and keep smiling.